Key concepts in histology. Definition and importance of histology in physiology. Histology is the study of the microscopic structure of tissues and their organization within organs. It serves as a foundation for understanding how cellular and tissue-level organization contributes to physiological function. Recognizing normal histological patterns is critical for identifying pathological changes, which are often the first indication of disease at the microscopic level. Basic tissue types and their functions. There are 4 primary types of tissues: epithelial, connective, muscle, and nervous tissues. Epithelial tissue covers body surfaces and lines cavities, playing roles in protection, secretion, and absorption. Connective tissue supports and binds other tissues, while muscle tissue is responsible for movement, and nervous tissue is involved in transmitting electrical impulses to regulate body functions. Epithelial tissue classification and specializations. Epithelial tissues are classified based on the number of layers and the shape of the cells. They can be simple, one layer, or stratified, multiple layers, and the cells can be squamous, cuboidal, or columnar. Specialized epithelial structures include cilia for movement, microvilli for increased surface area, and goblet cells for mucus secretion, all of which contribute to the tissue's function in organs like the respiratory and digestive systems. Connective tissue composition and role. Connective tissue consists of cells, fibers, collagen, elastin, and reticular fibers, and an extracellular matrix, ECM, that provides structural support and mediates biochemical interactions. The ECM's ground substance varies in consistency and is crucial in tissue hydration, nutrient transport, and mechanical resilience. Fibroblasts, adipocytes, and mast cells are key cellular components, with collagen being the most abundant protein, providing tensile strength. Muscle tissue, types and functions. Muscle tissue is categorized into 3 types: skeletal, cardiac, and smooth muscle. Skeletal muscle is under voluntary control and responsible for body movements, while cardiac muscle is involuntary and found exclusively in the heart, generating rhythmic contractions. Smooth muscle controls involuntary movements in organs like the intestines and blood vessels, contributing to functions such as peristalsis and vasoconstriction. Nervous tissue structure and function. Nervous tissue is composed of neurons and neuroglia, supporting cells. Neurons are responsible for generating and transmitting electrical impulses through the axon and dendrites, enabling rapid communication between different parts of the body. Neuroglial cells such as astrocytes and oligodendrocytes provide metabolic and structural support to neurons, crucial for maintaining the central nervous system, CNS, integrity. Tissue repair and regeneration. Tissue repair involves regeneration or the formation of a scar through fibrosis. Regeneration occurs when cells divide and replace damaged tissue with the same type, maintaining normal function. In contrast, fibrosis results in the replacement of normal tissue with collagen-rich scar tissue, often leading to functional impairment. The ability to regenerate varies among tissues, with epithelial tissue having a high regenerative capacity, while cardiac muscle is limited in this regard. Histological staining techniques. Histological stains are essential for visualizing tissue structures under a microscope. Hematoxylin and eosin, HNE, is the most common staining technique, or hematoxylin stains nuclei blue and eosin stains the cytoplasm and ECM pink. Special stains such as Masson's trichrome and periodic acid Schiff, PAS, are used to identify specific tissue components like collagen and glycogen, respectively, providing detailed information about tissue pathology. Pathophysiology of fibrosis. Fibrosis is the pathological formation of excess fibrous connective tissue in an organ or tissue, typically as a reparative response to injury. It is driven by chronic inflammation, where persistent activation of fibroblasts leads to excessive collagen deposition. Conditions such as pulmonary fibrosis or liver cirrhosis result from this process, leading to impaired tissue function due to stiffening and scarring of the affected organ. TGF-beta, transforming growth factor beta, plays a key role in driving the fibrotic response by promoting fibroblast proliferation and collagen synthesis. Antifibrotic medications and therapeutic approaches. Antifibrotic drugs aim to reduce excessive collagen deposition and prevent tissue scarring. Pirfenidone and nintedanib are examples of antifibrotic agents used in the treatment of idiopathic pulmonary fibrosis. Corticosteroids, another drug class, can also modulate the inflammatory response and reduce fibrogenesis by inhibiting cytokines like TGF-beta, which are implicated in conditions like pulmonary fibrosis. Thanks for watching. If you found this video helpful, don't forget to give it a thumbs up and share it with others. Subscribe to this channel and hit the notification bell so you never miss an update in the series that we are doing. For more learning resources, check the description below. See you in the next video.